regulation of translation by small RNA molecules. I've already alluded to this earlier when I was talking about the 3' untranslated regions of messenger RNA transcripts, and those can sometimes serve as a source of the small RNAs for gene regulation. But anyway, small RNAs or non-coding RNAs don't function as messenger RNA, tRNA, or ribosomal RNA. Some antisense RNAs are complementary to messenger RNA and function by base pairing, and that complementarity is what they use to shut down gene expression at the level of translation. Obviously, the transcript is already made, but then with a complementary RNA stuck to it, it can't be translated. So again, it may inhibit or enhance translation. Translation by antisense RNA looks kind of like this. So you have a messenger RNA that's with a prominent ribosome binding site there, just for ease of viewing. And anyway, on the left, translation will take place because the 30S ribosomal subunit can find the RBS, and the 50S can find that, and they form that 70S complex. Over on the right, however, the small RNA is made, and the protein HFQ associates with it, and that complex can then bind to a transcript and block the ribosome binding site, or Shine-Dalgarno, to inhibit translation. Some of the mechanisms used for global regulation, a very short list, and there's other things, but regulatory proteins, which I've already talked about, just transcription factors, like RSC, for example, are alternative sigma factors, which I'll get into in a minute, two-component signal transduction systems, which I've already mentioned, and phospho-relay systems. So the latter two are kind of overlapping. The two-component systems are typically phospho-relay systems. Here's a list of E. coli sigma factors. And remember that sigma is one of the components of the RNA polymerase complex in prokaryotes. And so you can see a variety of different sigma factors, like sigma F, for example, will associate with RNA polymerase and drag RNA polymerase specifically to promoters of genes involved in flagellum assembly. Another version of regulation is by nucleotides, which is pretty unusual, like guanine tetraphosphate, or PPG-PP, responsible for the astringent response in E. coli, or cyclic diGMP, which also regulates a whole bunch of things like virulence and motility and biofilm formation in bacteria. As far as the astringent response, through PPG-PP, the signal itself is synthesized in a variety of different ways, as indicated in this. You can see that either ATP or GTP can be used to make PPG-PP, or ATP and GDP with the concomitant loss of AMP can produce that as well. So here we're losing AMP, and then we lose another phosphate to make that. But anyway, just to give you a sense for how these things are formed, this signal is formed. So the stringent response text is here, and the actual diagram is here. And so this is really predicated on this protein rel-A. So you get, you can get, the amino acyl tRNA comes in this particular case. It's an arginine amino acyl tRNA. You get peptide bond formation, meaning this nascent polypeptide chain gets stuck onto the arginine, and then the ribosome shifts over. That leaves the A site open for the next amino acid. And so imagine it's a threonine. This is the codon for threonine, and there is no threonine. And so you have uncharged tRNAs that are not loaded with threonine. And as a result, that stalling of the ribosome would attract rel-A to bind, and then rel-A would activate the astringent response, the PPG-PP response. And that signal, the PPG-PP, as you see here, would either activate certain genes or shut certain genes down. And so it depends on what the gene does. So you would not be surprised to know that the upregulated genes might be genes that are responsible for making more amino acids. And the genes that are upregulated typically have this discriminator region here, this AT-rich discriminator region, whereas genes that don't need to be synthesized during this period of crisis in the cell would be downregulated. Their discriminators typically have a GC-rich region. And so again, the PPGPP would block this transcription complex in the presence of DKSA, which destabilizes it as well. As far as the cyclic diGMP goes, this is a great example. This is just some work that was done in a friend of mine's lab at the University of Loyola College of Medicine in Chicago. And so what the Weissig lab showed was that diguanylate cyclases and phosphodiesterases work together to regulate biofilm formation in Vibrio fischeri, a microbe that I work on. And so in this particular case, GTP molecules in the presence of this enzyme diguanylate cyclase are used to make cyclic diGMP. That's the signal, and that signal is what activates biofilm formation. And phosphodiesterases, on the other hand, cleave this molecule here and here to open it up and to turn the cyclic diGMP into PGPG. And so again, basically it's just, it's cutting one of these phosphodiester bonds. So you get PGPG. So imagine just cutting this phosphodiester bond right here. So again, the diguanylate cyclase activates the biofilm formation, and the phosphodiesterase represses it. And so in this particular set of panels, you see the wild type. Here's a colony of the wild type bacteria, nice and smooth. It looks like a little Nilla wafer cookie. And here is a representation of the biofilm formation in wild type. It's very minuscule. In this particular assay, we use a dye called calcofluor to stick to the biofilm. In this particular case, it's a biofilm made out of cellulose. And so the wild type doesn't produce a ton of it, so the diguanylate cyclase signal is not that strong, and so you don't get much, if any. You get a tiny little bit of biofilm formed in the wild type. But what my colleague Karen Weissig did was her and her lab created a mutant in this particular gene called BIN-A. So delta BIN-A means that they deleted the BIN-A gene. Basically what they did was they deleted a phosphodiesterase. And as a result, the loss of this phosphodiesterase led to a buildup of cyclic diGMP. And so in this mutant, the phenotype is a really pronounced band of biofilm formation, and the colony is very wrinkled. So that wrinkling also suggests that the bacteria have upregulated biofilm formation. Over here, in any good molecular biology experiment, you have to complement the mutant, meaning you add the gene back just to show that you can get a restoration of the wild type phenotype. And so that's what she did there. So again, the BIN-A mutant the BIN-A gene was then reinserted into the BIN-A mutant to restore the wild type phenotype. So again, that shows you how these diguanylate cyclases and phosphodiesterases work towards biofilm formation in a particular bacterium. Here's another example of that. So this is in Vibrio cholerae, where cyclic diGMP is an activator and repressor of two competing activities. So here's our cholera organism, and the organism makes a protein called VPSR. So when VPSR comes in contact with cyclic diGMP, that complex activates VPS. This is the Vibrio polysaccharide operon. And so VPS encourages biofilm formation. So that makes the bacteria stick to surfaces and encourages a sedentary lifestyle. At the same time, if there's a lot of cyclic diGMP, it will also bind to FLRA, this protein. And FLRA, cyclic diGMP, that complex will actually block flagella synthesis. And so you get a block of motility and an activation of biofilm, which should make sense, right? The microbes won't need flagella if they're pasted to a surface.